There we go. We're being recorded. So I was trying to look through the mind maps and I got through a couple of them and I thought, you know, if we were all in the same room in the computer lab as originally planned, it would have taken 10 minutes to go through and make suggestions and interact with one another and talk about it. And now, because it's this digital sharing, it's taking forever. It's taking forever. There was really a pretty clever little mind map shared on MindMeister, and we'll all get to see these things probably next week. I'm assuming Tuesday. And it took forever. MindMeister didn't want to give me access. It just became this cascade that what should have taken a few minutes in class, you know, ended up taking about 20 minutes online. So I think this has just been the lesson of this whole semester is everything takes twice as long, so you got to expect half as much. And I hate to say that because I really hate not giving you guys a better experience. So, but that's, you know, that's a me problem. That's a me problem. I have not, but I have not forgotten as well. I have not posted the plenary talks from the genetics meetings yet because the recordings are not available yet. As soon as they are available, I will post them. The genetics meetings, very interesting, are all online this year. Next spring, they are being held in San Diego. So I'm going to be in San Diego next year. So if I'm teaching bioinformatics, or if you're going to be here, you can expect there will be a week in end of March, early April that I will not be here because I am going to San Diego to the genetics meetings. I just need to go and see some real scientists in person. But the talks have been very good. They made an interesting decision on some of the talks. So instead of the meeting being for a week, they're actually, the meetings are going to go for the next 2.5 weeks. And basically they have half a day of talks in each day throughout the next couple of weeks. And the funny thing about them is that they are letting people give the talks in the time zones that they live in, because it's all on Zoom. Which means that I was at 11:30 last night watching a talk on chromosomal structure and genetic dissection of centromeric proteins, genes for centromeric proteins in the middle of the night, which, great talk. I was happy to do it. It was fun to interact with some international colleagues, but it's a very weird way to do a scientific meeting. So I guess that's kind of what we all have. I'm hoping that not all meetings are going to be like that. We'll actually be able to be in person. So this is about the time in the course, now that we've kind of got a little experience in mind maps, a couple experiences. I'm just putting my slideshow up here. Ugh. There we go. And I'm going to share the screen. Don't panic. Hang on. There we go. That should work. And this is really showing you a little more detail of how you can use our idea of mind maps and pathways to show what should be by now in your biological and science education, a fairly recognizable system. And in this case, this is something we all know and dearly love, is intermediary metabolism. And so again, I'm going to try from here on out, and if it feels like you're jumping into very ice cold water, I want to move away from the computation element for a couple of weeks, really to jump into biological applications. And that's really the purpose of the online talks last week, and a couple more to come in the next week or so. But I am going to, sorry, I'm looking off on my other slide here. I'm not being rude. I am going to skip through some of these slides. These will be available for you on our Blackboard. So you'll have access to not just the slide set, but also some of the websites we'll talk about in a little bit. So these are all of it, will all be available on Blackboard for you. And I am going to skip over some of these slides. First of all, they are complicated. They do not lend themselves necessarily to the Zoom lecture approach, but they will just cursorily go over them to remind you about certain elements of intermediary metabolism, but we'll jump pretty quickly into the use of mind maps and presenting pathways, which is really the main point, and using a pathway to describe what's really a rather boring topic in biochemistry. Most of you know we're talking about heterotrophs. None of the vocabulary here should be strange to you at this point. I'm sure that you went over them in Bio 1. If you've taken biochemistry, then you certainly have gone over these words in biochemistry and some of the ideas. So they should not be that strange to you. Basically, when we're talking about catabolism, remember metabolism itself is broken down into two separate pathways. The first is, of course, catabolism, and the other one is anabolism. So any guys remember which one's which? What does catabolism do?
Isn't catabolism the breaking down of molecules?
Absolutely. So catabolism, and I'm emphasizing the word, not really pronouncing it correctly, is breaking down of molecules to their more simpler metabolites. And of course, anabolism then would be the building up of more complex molecules from their simpler substrates. If you ever have trouble remembering, it's always, for me anyway, I always remember it and remind students taking biochemistry or learning Krebs cycle for the first time, talking about anabolic steroids. Thanks to our national obsession with sports, we all know anabolic steroids are taken by athletes who want to cheat and build up muscle. So it's the building up part that I want you to remember if you think about anabolism. Catabolic steroids, by the way, remember steroids play a whole host of roles in metabolism, and we'll see some of that a little bit later in a couple of slides. Of course, catabolic steroids are ones that break down complex molecules, and so we see a lot of the gluconeogenic enzymes, gluconeogenic, that is, they make brand new glucose, and they do so by breaking down starch and fatty acids. So that's another set of catabolic enzymes. So what are we talking about in the principal cycles for this in catabolism? Of course, glycolysis, the citric acid cycle, electron transport, oxidative phosphorylation, pentose phosphate shunt. These, if you recall from biochemistry, these are all part of the game, and you remember them probably. You may not remember all the bits and pieces, but you probably remember studying them and all the pain they caused you over the time you looked at them. They are always the bane of every biochemist's existence. As we mentioned, anabolism are the biosynthetic reactions. They are generally more complex than catabolism, and the reason is that they use energy. Usually, catabolic processes result in the production of energy, and especially from an intermediary metabolism point of view, the production of ATP, whereas anabolism utilizes ATP and utilizes it in order to couple smaller molecules together. So pretty straightforward. Again, you can read these in more detail. I'm kind of giving a summary of each slide. When we think about macromolecular synthesis, this is really the process. Why do we study it? Because this underlies the principles of growth. Okay, so how do we grow? How do we develop? If you think about organisms starting off as a single diploid cell, and then that cell continuing to replicate itself, undergo hyperplasia, that is increasing the number of cells, and then of course hypertrophy, the increasing in size of each individual cell, and then ultimately differential, or rather differentiation of those cells into different organs and organ systems. What we're really talking about is, and I do love this last sentence down here, that growth, just switching my pen here, if you look down here, notice my steady hand. Really, when we think about growth, it's represented as a cellular accumulation of macromolecules and the partitioning of these materials of both function and information, because remember, that's the role of DNA, it's informational, into daughter cells and cell division. So we really begin to see this. That's a great description of what growth is really about. I'm going to skip some of this because it goes into a little more detail about the biochemistry, but please do remember all of these biochemical adjuncts should be at least sounds familiar to you. Pyruvate, oxaloacetate, alpha-ketoglutarate, acetyl-CoA, succinyl-CoA, phosphoenol pyruvate. These are all elements of the Krebs cycle, and they're things that you should remember. Obviously, NADP or NADH and FADH, these are all elements that you should remember from either Bio 1 or certainly biochemistry. I don't know if anybody take physiology here? Do they talk about intermediary metabolism and physiology?
I took it, and I don't think we actually did. We talked about a lot of other stuff.
Oh, okay. Just curious. It's I think when I took although when I took graduate level physiology, this was like a huge portion of the course because it's such an important. They are such important mechanisms to understand the process of growth and differentiation. Just curious. I'm not going to get involved in this, but this talks about making ATP equivalents from cellular respiration, and don't worry about that. Here's where I really wanted to go. So all of those other slides basically have information which, even in the abbreviated reading that I gave them, an abbreviated lecture type, and I skimmed over a lot of the stuff. All of the information in those slides is contained in our model. And here's a great way that one can use the mind map to create a model of intermediary metabolism. And this was all created on, I did this on Xmind, so it's a slightly different software package, but you could do the same thing in any of the other packages that we've been looking at. And basically, you've got up here at the top, notice up here, we've got obviously plants, because we're doing photosynthesis. So here we're capturing smaller molecules and using energy from the sun to create carbohydrates. Now, the fun stuff comes when we look from, these are all our autotrophs, and notice it's really very simple, but contains the essential elements of the previous couple of slides. And of course, what happens to these complex carbohydrates? They're catabolic processes, anabolic processes. Notice here are all of our situation with intermediary metabolism. So we go through here, obviously breaking down all of our products into CO2 and water. Here are all the physiological intermediates. And then anabolism, where we're building up. First, we have to build the basic building blocks, and we do this from carbohydrates, create amino acids, nucleotides, fatty acids, and then ultimately macromolecular synthesis, in which we're really talking growth and differentiation. Now, notice that if depending on where we're thinking about, especially eukaryotic organisms, what begins to happen if we're talking bacteria, okay, then basically all bacteria really need are carbohydrates and a nitrogen source, okay, and certain minerals. So they do not, prokaryotes do not require individual amino acids, nucleosides, fatty acids, etc. They have the machinery, and by machinery, I mean biochemical machinery, to create all those things basically from carbon and water. As we get a little higher up the tree of life, and I know talking about higher up the tree of life is a little speciest, but as we move a little bit further up the evolutionary scale, we begin to see the appearance, especially in kingdom Animalia, where individual nutrients become essential. And so, for example, in the case of any vertebrate, there are individual amino acids that actually have to be consumed in a diet. Same thing for fatty acids, okay? Some have to be included in the diet. We do not have the molecular machinery to synthesize the individual components of what we would consider to be the macromolecules of life just from CO2 and water. And I'm sure this is something that's pretty obvious to you, but certainly something in the context of our big model here of being important to differentiate. So if I were going to do this this year and redo this, I would probably include in this section in here, I would probably begin to include another aspect indicating certain organisms which have essential nutrient requirements in order to make our buddies, proteins, nucleic acids, fatty acids. And of course, what do we do with those? Well, we're building membranes, organelles, cell walls, and then eventually, that's an arrow, organs and organ systems. So you can actually do a pretty fair job just by using simple mind mapping software to create fairly complex ideas and communicate them very quickly. So here's a great example. I like that one quite a bit. You can do the same thing on a more molecular level. Here is more of the chemistry of those of you, again, who took Bio 1 with Mr. Fergione. probably go crazy thinking C6H12O6 plus 6O2 gives you 6 CO2s and 6 waters. And of course, if you wanted to get more complex, you talk about what happens to all of the electrons. So we're talking about the stoichiometry, including NAD and FAD. So we really have all of this new information. Note that we can't predict the actual stoichiometry, that is the number of ATPs that are actually created from the chemistry itself. But really, this is the end result of biological adaptation. So it's kind of numerically based on the reaction points of the enzymes that exist in different creatures. There was a great paper came out about 4 years ago where they actually were able to measure very specifically the number of ATPs created in this case. And what was really interesting was it was just over 36. So it was really, really kind of cool to see an experimental number come so close to the theoretical number. But again, fairly complex stoichiometry. So is there a way that we can use our graphical analyses to show this in a little bit more understandable fashion? And of course, as you may have guessed, we certainly can. And here's a classic look at the phosphofructokinase and fructose biphosphatase. And here we see the classic metabolism of fructose through here. So we really have a great look at both. We can include pictures of the actual molecules themselves. All right, pretty neat. And then we can even include down at the bottom our net number of ATPs in this part of the substrate cycle. We can also, if we wanted to, show the unidirectionality of fatty acid oxidation and fatty acid synthesis. Again, showing, why is fat more energetic than carbohydrates? And of course, you could see the answers up above here. We got 35 ATPs down here. And we use 63 to actually construct fatty acids. So you can see that the metabolism or the catabolism costs much less than the anabolic process. It's pretty cute. Gives you an opportunity to make very, very rapid comparisons. I want to show you another view. Again, let's move away from the molecular for a moment and let's look at where do these processes occur. And again, thinking predominantly vertebrates, again, I tend to be oriented toward vertebrate organisms. All of these processes not only occur just in cells, but very specifically occur in particular tissues, and those tissues exist in particular organs. So here is, of course, a great mind map showing you an organ view of metabolism. And you can do this mind map just by looking up in the literature. This actually was created by an undergraduate research student of mine. And basically, we really look through basic nutrition and physiology texts to come up with this idea. And you can kind of see, then, of course, she had a ball making little pictures of hearts and brains and eyes and livers and, of course, muscle. And you could see in each case the relationships among and between the metabolites within each of the individual organ systems. As you probably know by now, glucose is the primary provider of energy in the brain. certainly, see if I can, well, that's kind of an arrow. And also in heart tissue. However, they are also able to metabolize energy from ketone bodies. So where do ketone bodies come from? Well, you can follow the line back and see that ketone bodies are produced in the liver. The ketone bodies are a result that occurred during heavily fasting states, especially amongst individuals who are not eating carbohydrates. I should point out that this is very popular these days. In the term, you've probably heard people going on a keto diet. Okay, they're basically trying to eliminate carbohydrates from their diet. This is a bad idea. Okay, it is not good for you know, you want to drive your body into using fat. That's the principal concept of it, right? Which is great if your liver is processing fat and breaking down fat. However, when your brain is in glucose deficit, the ketone bodies are actually produced by metabolizing fat in your brain, in your heart, in your liver. And of course, here's the bad news. Anybody know with regard to your brain where all the fat comes from? Where's all the fat in your brain?
Isn't that the axons?
Yes.
The myelin sheath, right?
Myelin sheath. That's
Yes.
exactly right. And do you remember, Cass, what's the function of the myelin sheath?
Oh my gosh, I literally just took neurobiology last semester.
Let me rephrase the question. Okay. What's the purpose of when you plug a lamp into a wall socket? Why is the electrical wire covered in plastic?
To keep the electricity in.
You got it. And the myelin sheath?
Keeps the neural impulse on track.
Absolutely. So if you get rid of the myelin sheath, you're in trouble.
Yeah, that's not Parkinson's. What's the other one? Where it's a degeneration of the myelin. MS.
MS, very possible, although not involving ketone bodies, but you're absolutely right. MS, Lou Gehrig's disease, there are a handful of myelin related diseases. There are a handful of them. But why intentionally put your body into a position of digesting and getting rid of fat in very important neural tissue? Generally, folks who have died on some of these extreme diets have died principally because of neurophysiological defects, usually in the heart and or the brain. So really, and imagine what happens if the impulses in the neural impulses in your heart, rather neuronal impulses in your heart, get interrupted. Well, your heartbeat gets interrupted. So anyway, just a brief little vignette that can be seen fairly easily once we put everything into the concept map. We really can see this, so a lot of variations in here. You know, another thing that you could see that you could immediately begin looking at, for example, is protein metabolism. So to just veer away for a moment from glucose, actually, I wanted to cut that off there. You can really see the relationship between liver and muscle tissue with regard to protein nutriture. Remember that the largest pool of free amino acids in your body, in any vertebrate's body, are actually located in your muscle tissue. It's not in your blood. Okay, it's stored in your muscle tissue. This is where free amino acids reside. And you can kind of see that here. And you can see the relationship by following the line between the liver, and muscle. So a lot of fun things you can talk about by having the conceptual map, the mind map, concept map, etc., put together. So really kind of cool. Let's see, I'm going to, these mostly are talking a little bit about energy metabolism. What's the reservoir for energy? So let's say you do stop eating glucose. What happens? Skeletal muscle is okay because there is glycogen in all muscle tissue, very common, broken down into bits and pieces. Of course, the preferred substrate is fatty acids. Why? Because we saw on a previous slide, fatty acids result in many more ATPs. So really kind of a nice way we can have a tabular presentation of looking at what the energy reservoir is, what the substrate is, and then of course, what kind of resource is exported for energy. There's some great information here about what happens in the brain. The brain is really pretty impressive in terms of its oxygen consumption. This is why sitting down and reading will actually cause you to lose weight, depending on how many bags of chips you eat while you're reading. But it has 20% of your oxygen is consumed by brain. Really quite remarkable if you think about that. Your brain has no fuel reserves. There really is nothing there. So if you begin to starve yourself, where does the energy to carry on metabolism, where does it come from? Well, as you begin to fast, or in the keto diet, for example, your glycogen reserves are depleted. Again, where's glycogen? We can go back to our picture, but one of the reasons I pointed it out was we have glycogen located in the muscle tissue, also in the liver, and then we begin seeing the accumulation of acetyl-CoA so that we can produce energy from citric acid. And the problem, of course, is the brain can't use it. So where does it come from? It comes from ketone bodies. Of course, as you might imagine, these complex biological issues are rather easily put together in an individual pathway diagram. So this is less of a mind map and more of a pathway. Again, you can use mind map software to create these, or you can do them by hand, or rather simply in, for example, PowerPoint. And you could show the relationship and the conversion of hydroxybutyrate into acetoacetate, acetyl-CoA, and then ultimately use in the citric acid cycle. How about another tissue? Again, muscle tissue, skeletal muscle, about 30% of the total oxygen consumed by the body at rest. And we are primarily geared in muscle tissue, as you might imagine, to the production of ATPs. That way we can keep going, keep moving. I'm going to skip through a little bit without getting bogged down into the physiology and calcium metabolism in looking at something like the utilization of creatine and phosphocreatine in muscle metabolism. So these are all important elements of muscle metabolism. Also, muscle fatigue. You guys have all said, oh, you got to keep going, to the burn, right? They say when you exercise, we've all done these exercise programs, and they say you have to feel the burn, okay? Anybody who's run or done any work in lifting and you feel the burning sensation in your muscles, there's really a little bit of a misunderstanding. Everybody assumes that the burn is because your muscles quickly utilize all the oxygen reserves. So the minute you're done with oxygen, what happens? Your body shifts into, your muscle tissue in particular, shifts to anaerobic respiration, which in muscle tissue, if you can't digest glucose and go through the citric acid cycle, it only goes through glycolysis. And in muscle tissue, the final metabolite or proton acceptor is actually lactic acid. So there's lactic acid buildup. A lot of people think that a lactate itself is responsible for the burning feeling in muscle. And that's not true. The problem is there's a decline in pH in the muscle, and it's actually the decline in pH due to protons that are generated in glycolysis. So it's really a much more complex thought. Of course, easier to show in the transamination, which allows us to get pyruvate and alpha-ketoglutarate from both alanine and glutamate to amino acids. So we actually have an enzymatic process where we can generate energy. Note, here's pyruvate, elements of the citric acid cycle here. So we have this accumulation of proteins. So what I did want to show you was, where can you find these things? So let's say that you're interested in putting together a concept map regarding any element of metabolism. I use this as an example because everybody should be familiar with intermediary metabolism by now in your biological education. But of course, there are a lot. Hold on a minute. There we go. There are quite a number of metabolic systems and metabolic pathways. And what I'd like to do is show you two places where you can get a tremendous amount of information to utilize in a path diagram if, for example, as one of the example projects in previous years was looking at ethanol metabolism. How can you get at ethanol metabolism? And how do you find the overview of the metabolic process in one place, as opposed to trying to go to a biochemistry textbook and look up alcohol dehydrogenase and what happens when alcohol is dehydrogenated, and what kind of metabolites are formed, and where's the effect, what organ system do they exist in. There should be places for you to find this. And it turns out there are several, but there are two that I'm going to share with you. The first is really one of the top ones, and this is a pathway software that is provided by the European Molecular Biology Group. And you could see, again, this will be in Blackboard for you to find. It's pathways.embl.de, okay, DE. And you can actually go through, and they've got wonderful maps of metabolic processes looking at metabolism itself, which is really everything we've been talking about to this point. What about pathways that involve antibiotics? You ever wonder how antibiotics work or are synthesized? Here you go. Secondary metabolites, and also what's different between when we talk intermediary metabolism, very often we're talking, as I mentioned, eukaryotic metabolism, certainly vertebrate metabolism, but of course you can look at microbial metabolism as well. So let's just take a quick peek that you can display. And Here is the overall, don't you love this? Here is the overall pathway map for all metabolic pathways. And if you poke in here, now I'm going to turn away so I can see the screen a little bit. Again, I'm not being rude. Well, actually, maybe I can move this over to my other screen. Hang on. But you can kind of see, and I'm going to enlarge this just to make it a little easier to read. You can see very quickly all of the different elements of carbohydrate metabolism, amino acid, energy metabolism is kind of what we've been talking about. Lipids, terpenoids on the left. And by the way, see that big old circle in the middle? What do you think that is? Citric acid cycle. And if you ever wondered why such an emphasis on citric acid cycle, just look at its position among all of the different pathways and how so many of them interconnect with the citric acid cycle. If you know the citric acid cycle, then basically you've put yourself in a position to understand nearly all of metabolism itself. If you were looking at it and you say, boy, there's another cycle here that's really important. And I don't know if you could see it, so I'm going to enlarge it. Another cycle extremely important amongst all of the biosynthetic pathways.
I don't know if this is for everyone. Sorry, Professor, but I only see a sliver of your screen.
Oh, you only see a piece of it?
Yeah, it's like
poop. All right. Sorry about that. Glad you said something. Is that better?
Yeah.
Okay. Sorry about that. Thanks.
Yep.
I was trying to move things around so I could like look at the camera but still see the slide. So at any rate, let's go back just so you can see where we were. If you look at the overview of the entire cycle, of course, you see here's the importance of the citric acid cycle. But there's another circle that's also extremely important, and that's over here. And if we look at that one, I'm going to make it bigger so I could drag it around to show you. It is, of course, the urea cycle. And you may recall from biochemistry, the urea cycle is extremely important because it deals with nitrogen. Remember, citric acid cycle is predominantly about what? Carbon, hydrogen, oxygen. That's why it is fundamental. The urea cycle has to do primarily with our other big biologically related atom, and of course that is nitrogen. And so if you look at all the yellow, and this is brilliantly color coded, so we can see all of the yellow has to do with nitrogen metabolism, and especially among eukaryotes, what's the really important part? Amino acids. So amino acids and protein. So you really get a sense, looking at the total map, carbohydrates are in purple. We see nitrogen and amino acid metabolism in yellow. Okay, everything is color coded, so you can kind of see everything that you'd really be interested in. Really remarkable that this occurs in all cells. And how else is there a better way to illustrate or to communicate these processes than via a concept map? That really boils it down right here. Now, I do want to show you one more pathway, and this is an absolute phenomenal resource. And this is the Psych database collection. It covers all of those metabolic systems that we saw in the previous slide, and really talks about how they are individually integrated with all of the classic bioinformatic databases. Which databases? Okay, the protein databases, the gene databases, the genomic databases. And so you can kind of come up here, and one of the great things about this is you can come up here and search on any particular pathway. So if, for example, you are interested in ethanol metabolism, you can do a quick search for ethanol metabolism. Notice we have huge number of results here. And we could say, well, let's look at ethanol degradation. And I'm just going to look at the first one just to make life easy. And what it does is it automatically puts you into the primary metabolic pathway for ethanol degradation, alcohol or ethanol dehydrogenase, how it functions. It depends on NAD. Of course, the conversion from ethanol, if you took health in high school, you probably know when you drink alcohol, it turns into an aldehyde and you get acetaldehyde, and the acetaldehyde in your body is converted into acetyl-CoA, which can then enter glycolysis. And notice we have all the links here, so we can continue to superlink over to, there we go, our handy dandy pathway of glycolysis and the citric acid cycle. And it's all plotted out here for you. Plus, all of the individual metabolites are hot linked to show you not just the compound name, but also, for example, how gene expression of the different enzymes are controlled. So really, this is a fantastic resource. I'm going to show you next week one of my little projects. And I am going to change my organism database here. And since it is the Drosophila, come on, oops. This is the Drosophila genetics meetings this week. I'm going to change my database. And what I'm going to look for is a compound called drosopterin. Come on. Ah. I don't know why it doesn't show there. Yikes. All of a sudden, why is this not working? All right. I'm going to change my search term here. There we go. So basically what happened is I'm looking here for a gene called brown. And unfortunately, I'm not finding what I want. Well, I will show you the mine. Just one second. One more thing I want to look up here. Why is it being really cranky? Oh. All right. I'll show you. I'll show you the. I'm obviously forgetting the correct search term here, but I'll show you the one that I built next week, where we're looking at metabolism of different compounds to create eye color in Drosophila, and how we use our mind mapping programs along with creating pathway diagrams to indicate all the different mutations that can occur along the way to creating eye color in Drosophila. So again, another way that we can focus on using our graphical techniques to present a rather complicated, in this case, metabolic process. So hopefully you get a sense from the handful of the graphics that we've shown is you can do everything from very general, going from organ systems, for example, and looking at metabolism within the different organ systems, to very, very specific molecular relationships and biochemical pathways, down to the point of actually drawing out the biochemical structures. And in each case, whether from the very general to the very specific, in each case, our ability to create these concept maps or pathway diagrams helps to communicate the information that's contained in them. So I will get, over the next couple of days, I will finish commenting on everybody's mind maps. I'm about halfway done, so I should be able to get moving pretty quickly on them. And I'll also get those talks posted from the genetics meetings as soon as the recordings are made available. And this is a perfect stopping point. So continue, please, to work on your ideas for your projects and your mind maps, and you should be kind of generating. Don't feel like if you think you'd rather do something else, you can't change. We got, even though we do have a month left, there's a lot of time to make modifications and thoughts. And what I want to get to the point is not next week, but probably the following week, of actually beginning to put together some network diagrams, get back to using our concepts from our mind maps and path diagrams, and using R to create network diagrams from those outlines, and really, really hopefully get you a little bit deeper into your project. Okay? It's supposed to be 80 degrees tomorrow. Get to the beach. Take care, everybody. Hope everything goes okay for the have a good weekend.
Hey, Professor.
Yeah.
Professor.
Yep.
Hi. I had a question. So
Yeah.
I'm still having problems sending you my mind map. I don't know. I tried to upload it on Blackboard, but it wasn't showing up. Then I tried emailing you a link. But
Yeah.
I
Did
think
you get my comments on your mind map?
no, I don't think so.
Yeah, log
No.
back into MindMaster. I was wondering if it would send you an email that I made comments.
Oh, okay. Let me. I'm logged in now, and I don't
Okay, go ahead.
see. Let's see. Yeah, I don't really see any comments.
All right.
But you were able to see the mind map.
Yep.
Just
Yep.
clarify. Okay. Okay.
Yep. And I definitely wrote comments. Let me see if they're still on my end here. And now it's not showing it as being here. Hold on a minute. I'm going to try to, hang hang on. Okay. I think you need to send it to me again. Something changed because I had it and now it doesn't show up. Wait a minute. Let me try another one. No, I just got bumped out of it. I can't access it anymore.
Okay. Do you want me to just send you the link?
I think what you can do is somewhere you should be able to share it with me.
Okay, let me see.
So in other words, it's not a link. Yeah, I keep getting an access denied, so something must have changed.
Let me try. I
Dr.
think there
Barbato,
is-
Yeah,
if we have our mind maps on MindMeister, do we share it with you through like MindMeister with the share button and send it to your email?
that's the easiest way to do it. But
Okay.
however you want to do it's fine.
All right, I shared it. I don't know if you got it, but I just shared it.
Yeah, I just shared mine as well.
All right, let me check here. Hang on. Inbox, let me check. Okay, Dan, here's yours. Oh yeah, Dan came through fine. And here's Amica, open map. Yep, I got him now.
Okay, great.
All right,
Good,
great.
good to go.
Sounds good.
I'll
Thank
go
you.
through, and I think, Alex, if you look, I know I made comments on yours, and I still see yours here in extinction.
Yeah, I saw your comments.
Oh, okay, good.
Yeah.
Good. Yeah, something weird happened with yours because, like, I was entering my comments, and all of a sudden, this big thumbs down button showed up, and I was like,
Yeah, I don't know. I think you could like
"No,
rate it.
that's not what I was saying." I didn't do a thumbs down. I was just
It's all good. Thank you.
all right. You guys have a great weekend. I'll talk to you.
You too.
Take care.
I wanted to ask you a question real quick.
Yeah. What's up?
Yes. Since we didn't really get to like talk to you about stuff, I have my mind map like I don't know, like sort of done, like I'm starting it, but I wasn't sure if it was too broad a topic. I have like the center piece being like reproduction in the kingdoms, and then
Cool.
branches out from all of the kingdoms is like each of the reproduction. I wasn't sure if that was like too broad. Like I wasn't sure really what my topic should be focused on. If I should be
I
focused
think the
on more.
beauty of it is, I think the beauty of it is creating the mind map can help you. You can be as broad as you want, or you can be as specific as you want. And the mind map can help you focus by developing it and looking at the individual categories.
So do you think I should do like a broad overview of all five, or sort of just like
Well,
focus in on?
However
specific?
you started, why don't you share it with me and let me take a look?
Okay.
Okay?
Yeah, I'm doing it on FreeMind. So how do I share it with you on this?
I'm not sure. You might have to send it to me.
Okay, I'll just like send a picture of it.
Yeah, that'll work.
All right, thank you.
All right, sounds good. Take care.
Bye.
Bye.